Hi, thank you for joining me today. My name is Carolyn Peterson, and I'm a Senior Manager of Discovery Services here at Thermo Fisher Scientific. Today I'm going to talk to you about our stem cell services and how we can be an extension of your research team. The CellModel Services is offered in a workflow fashion, where clients can come in at any point, in and out of any process during the workflow. Starting with reprogramming, customers can provide patient-derived samples where we can reprogram them using our cytotune virus, as well as move into engineering using TALs or CRISPRs, differentiation using our differentiation medias, and assay development and screening using our many platforms offered throughout Thermo Fisher Scientific. Also, during this process, we offer full characterization of any cell line that you may have that you can provide to us, or we can begin with our own cell lines that are offered through our catalog. As mentioned, you could choose a single step or the complete workflow. At Thermo Fisher Scientific, we have built an infrastructure to manage custom projects seamlessly. We have a project management office as well as a custom biology R&D team. These two teams work together to deliver your projects to you on time at the quality you need. The project managers act as a single point of contact before, during, and after your project, as well as schedule all meetings and provides frequent proactive communication. They also track customer satisfaction, and all feedback received is taken seriously and used to improve our processes. The custom biology team is a group of dedicated individuals focused on customer-driven projects. They provide anything from custom assay development, cell engineering, screening, and profiling, and they use the entire Life Technologies and Thermo Fisher Scientific offering to craft solutions for our customers to meet their needs. Why should you choose Thermo Fisher Scientific to be your services provider? We have a proven track record for existing services. We've been offering services for over 10 years in screening, profiling, and custom biology with a very high on-time delivery and success rate. When you receive a stem cell service from us, you have the ability to purchase the cells or products used in the service. And at each milestone, we will provide protocols for each step of the process describing the products needed and how we did it if you choose to bring this work in-house. We also have demonstrated our stem cell experience by our collaboration with the Parkinson's Institute and application notes that are available where we've reprogrammed patient samples into iPSCs, differentiated them, as well as edited them using our Talon technology. We also offer Life Lab training courses. These are training courses available to clients where they would go to one of our labs in one of our locations and receive training on how to culture cells and use our products. We also have how-to videos and protocols on the web for all of our products. Now I will take you through each step of the workflow. The first step I'm going to start with is reprogramming. For the reprogramming workflow, we offer 4 basic milestones. Please keep in mind that these milestones are suggested milestones, and we can customize based on client need by adding or subtracting milestones or certain characterization steps as necessary. First step is the material receipt, where we would receive your materials, your patient-derived samples, and we would perform our QC, so karyotype, mycoplasma, and human pathogen. At this stage, we will provide a milestone report letting you know that we're going to the next phase, which is reprogramming. We will be using our Cytotune IPS 2.0 Sendai programming kit, which I will discuss a little bit on the next slide. We isolate 12 clones and expand the top 5 clones. Again, a milestone report is returned to you at this stage as well. Generating the seed bank, expanding and cryopreserving the top 3 clones, and then karyotype analysis on the top clone. At any stage, you can receive milestone reports as well as intermediate materials. The final stage where we generate the master bank, this is a fully characterized clone that we return back to you by cryopreserving up to 10 vials or more, depending on your needs, and verifying karyotype, viral clearance, and pluripotency by fax. The CellModel Services team recommends using Cytotune IPS 2.0 Sendai reprogramming kit to make iPSCs from somatic cells. This kit is currently available in the catalog and can be purchased for in-house use. The reason why we use this kit is it's very easy to use and it requires only one application to obtain successful reprogramming. It has an increased efficiency 200-fold higher than lentivirus, and it clears rapidly as it uses a non-integrating Sendai virus. The next step of the workflow I'd like to discuss is differentiation. Currently, the only protocol we offer is a differentiation into neural stem cells. As we add more products to our catalog, we will also offer differentiation to other cell types. For the differentiation to NSCs, we offer 3 suggested milestones. As with reprogramming, these milestones can be customized to your needs, and different things can be added or subtracted based on your final requirement. Our first step is material receipt. So if you will be providing us an hpsc line, we will perform a karyotype, mycoplasma, and human pathogen as a QC. If the material was generated by us here at Life Technologies, the IPC line, we can skip this step. The next step is a differentiation into neural stem cells using the Gibco PSC neural induction media. The final milestone is generating a master bank and expanding the NSCs to passage 5, cryopreserving the vials, as well as doing a full characterization. As with our reprogramming tools, the differentiation tools are also available in our catalog. The Gibco PSC neural induction media is available as it enables the induction of ESCs, iPSCs to neural stem cells with minimal time and effort. The rapid approach of 7 days and efficient neural induction greater than 80% from hpsc monolayers. Current methods take 14 days with much lower efficiency. The media produces high-quality NSCs with robust neural gene expression of SOX1, SOX2, and PAX6. NSCs with potential to differentiate to neurons, astrocytes, and oligodendrocytes. The next step of the workflow is assay development and screening. We offer a wide array of assay platforms that you can use to develop assays and screen in your cell models. I've listed a series here that are just examples, but we have many additional platforms that can be used to develop assays and screen in your cell model of choice. The assay development workflow will really depend on your cell line and the assay that you choose. Again, basic milestones that could be fully customizable, but these are the suggested ones. Receiving materials, whether they come from us or they come directly from you. What assay are we going to develop, and getting those conditions right, as well as validating that assay by testing client compounds or plate-to-plate and day-to-day variation. Again, this is fully customizable. The milestones can meet the needs of your assay and the outputs that you desire. Characterization can be done at many steps of the workflow, and we offer a variety of services to meet your needs in this space. The one I'm going to focus on is the tackman scorecard assay. Here is a general menu of characterization services we can offer at any stage of a project: protein marker expression, cytogenetics, and gene expression analysis. Today I'm going to talk more about the tackman hpsc scorecard panel as a characterization service that we can run for you. The tackman hpsc scorecard service has 3 major steps. The first step would be receiving your materials. We accept RNA, iPSCs, EVs, or differentiated cells. For the next step, if you do not send us RNA, we would prepare the RNA and cDNA samples and run the hpsc scorecard assay. From this, we would upload the data to our analysis software tool located on the lifetechnologies.com website. The final delivery is a data report that contains a result summary, scores relative to the reference standard, expression plot, correlation plot, and box plot. The tackman hpsc scorecard panel is a qPCR assay that integrates gene expression analysis with bioinformatics for simple, easy-to-interpret results for your cells. It confirms pluripotency, determines trilineage differentiation potential, and optimized directed differentiation protocols. Here is a table that demonstrates the sample requirements and timelines for services. Listed are the sample type, the amount of and method of prep needed, and the timeline to receive the report. As you can see, if we receive purified RNA or iPSCs, EBs, or differentiated cells, the timeline to receive a data report is 1 to 2 weeks. The longest timeline is providing undifferentiated cells that require EB formation. Thank you for listening to this executive overview of our services offerings. Each of these services has additional slides that demonstrate case studies. If you would like more information, you can visit us at www.lifetechnologies.com/cellmodel, or you can send us an inquiry. Directly reach our project management team by emailing us at discoveryservices@lifetech.com. And if you require technical support on any products already purchased, you could reach us at stemcell-help@lifetech.com. Thank you again for listening. All products and services reviewed in this slide deck are for research use only and not to be used in diagnostic procedures. Life Technologies is a Thermo Fisher Scientific brand. Tackman is a registered trademark of Roche Molecular Systems, Inc., used under permission and license.